Thanks very much, Amanda, and thank you for staying this late. We're going to talk about the evolution of complex genetics as part of a larger program looking at the evolution of complexity throughout life. A couple of previous speakers have talked about this. I'm not going to talk about the sort of transitions leading to complex life. But we're looking at complex function, particularly. So how do complex organisms such as ourselves evolve? And not in terms of specific anatomy, such as, how does the human eyeball evolve, but how does things like imaging vision evolve? And imaging vision has evolved multiple times through different independent routes, things like powered flight. Lots of these complex functions have evolved independently multiple times once you've reached multicellularity, which is a completely different issue. So we want to know why has this happened, and therefore could it happen on other worlds with a life capable of doing that? What's the requirements? In order to have life with complex function, you have to be able to have differentiated cells that can perform lots of functions, have specialized molecules within them, and they have to be put in place in the right place and at the right time. And in developmental programs, they have to then be taken away again at the right place and the right time. So this is a very complicated control process, and that speaks to there being a complex genetic program underneath that to make that all work. So we've been asking, is the evolution of this complex genetic program, the capability to evolve a complex program, itself a bottleneck in the evolution of complex life? Complex genetics is pretty much restricted to the eukaryotes of this sort of complexity. So eukaryotes tend to have big genomes. This is quite an old plot. This is all the completely sequenced genomes I could find in a database. These green guys here are the eukaryotes. Along here we have genome size in log scale and megabases. We're up to gigabases here. These are huge amounts of information. And eukaryotes can develop and have developed independently multiple times very complex developmental programs. By contrast, prokaryotes, and I don't apologize for lumping everything else together in prokaryotes, I know it's old-fashioned, there you go, tend to have much smaller genomes, and when they do develop multicellularity, it is simple, it is limited, a very limited number of cell types, and it is quite often facultative, so they can switch between unicellular and multicellular states. You and I can't do that. Well, I can't. But the genomes do overlap in sizes. There's not a distinct size gap where you have to jump from prokaryote to eukaryote, and they also overlap in chemistry. And this is something we spent rather too much time illustrating, but I'll try to be brief. If you look at all the chemistry that's involved in how genes are controlled in an organism. So you have DNA binding to RNA, RNA binding to proteins, proteins binding to DNA. There's a whole zoo of interactions, and you can modulate any one of these by reacting them with different chemicals, and then each of them can control different steps in the process of activating a gene. So from initial transcription all the way down here to breaking down the protein at the end of the process. And you ask, what combinations actually occur? Okay, so you can have a targeting moiety, a protein molecule over here, and it binds to something else, and then something happens. So you've got targeting, targeted, and what happens. And if you look at what happens in the eukaryotes, the answer is pretty much everything. And you look at what happens in the prokaryotes, and the answer is pretty much everything. All the combinations are there. There isn't a distinct chemical pattern of this. The chemistry is similar in all the major domains. Differently distributed, different emphasis, but there's not a fundamental chemical difference. And these are independently evolved. They are not the same, and we know they're independently evolved because they're completely different sequences, different structures, and so on. And so our hypothesis, which we developed, is that the inherent difference is the control logic, and that eukaryotes, us, have a gene structure that is by default off. If you add a gene to a eukaryotic genome, it will by default not do anything, and you have to do a lot of work and activity to turn it on. Prokaryotes have a default on logic. If you add a bit of DNA to a prokaryotic genome, it will tend to do something. There's lots of evidence. I won't go into this. We've written a very long and, to be honest, rather boring paper about this. I should emphasize this is a style of control. This isn't absolute. Okay, so of course you can find examples of gene systems in prokaryotes that are default off. Of course you can find ones in eukaryotes that are default on. But it is a general style of control. And our hypothesis, which I'm not going to justify today because we don't have time, is that this is related to why eukaryotic genomes can be amplified through gene duplication and made more complex. It pre-adapts you to be able to do that. Okay, so is that true? Well, we can't go back and with a time machine. You know, the snowflakes thing is great, but we'd have to run a yeast snowflake experiment for 1 million years to test this. That's not really practical with a 5-year project grant. So we thought we'd model it. And what we want is a model of the sort of complexity that occurs in genetic systems, in evolving genetic systems, which are spaghetti code. And if you look at the sort of control maps that biochemists like to put up on their wall to show how mature they are complete spaghetti code control systems. There's no layers, there's no modularity. So we wanted to model that. And there's a lot of feedback between gene products and gene expression. So this sort of highly abstract network formulation didn't really work for us. But equally, we didn't want a model of detailed chemistry, this sort of thing down here, proteins bind to DNA, because if you try to do that on a cell basis, then well, that would be too complicated. I wanted something that looked like the classic undergraduate textbook diagram of a Jacobian mono control model. For those of you biochemists, something that looks like this. Okay, so that's what we built. So this is the model, and it's conceptually quite simple. I'll tell you about the coding later. That's a disaster. So we have a population of organisms. And yes, we do have 5 organisms at the moment in the model. And those organisms contain genes, and those genes can be active and produce transcripts. We need selection in this. This is an evolutionary model. So for an organism to be fit, those transcripts must match an environment. And there are positive aspects of that environment, things it must do, and negative aspects, things it must not do. If it's just positive, then you just select something that makes everything, and then it's one. Okay. How do you tell whether you've got a transcript? Well, the transcription of each gene is controlled by regulatory elements. And you've got positive regulatory elements and negative regulatory elements. Positive, turn it on. Negative, turn it off. A note, we've only got one type of sequence here. We haven't got translation built into this. I thought of doing that and then thought it's too complicated at this stage. So if you believe in the RNA world hypothesis, this is a sort of RNA world type genome. How do you control the genes? Well, pretty straightforward. We've got positive elements and negative elements. If the number of active positive elements outweighs the number of active negative elements, it's on. If it doesn't, it's off. Key bit is what do we mean by active. We want feedback between what the genome does and how it controls its genes. So a genome produces a sequence here, a transcript, and we ask for each regulatory element here, does that match a transcript that's being made at the moment? So here's a gene, 2 on, 1 off. This one is produced. Let's say the transcript of this sequence. Here's another gene. This regulatory element has sequence ACA. That matches that bit there. So that's on. This one is CCC. That doesn't match anything here. That's not on. This is BC. That's not on. This is C. That matches that one there, so that's on. And you do this for all these regulatory elements in all the genes, matching all the transcripts, and that tells you whether that particular gene is on or not. 2 things to note. This is a model of regulatory control of genes. It's not structural. We're not saying this is an enzyme or something does something clever. So it's a model of gene regulation. And secondly, this is sort of a bit obvious, but I'll say it anyway, because it's late in the day and your blood glucose levels are dropping. If your regulatory sequence here is short, it is more likely to match something here. So if you have a short regulatory sequence, it's more likely to be active. Okay. Lots of variables in input for this. My goodness, you can have fun with this. So we're not going to go through all that. We're going to look at some summary statistics of what the output looks at. And there are 2 things we're going to focus on, one in particular. If you have a lot of these negative elements, and you could lose elements through selection, they can be deleted. So if an organism decides it only needs 1 regulatory element, that's absolutely fine. Eventually it'll get that. Lots of negative elements compared to positive suggest you've got a sort of default off state. They'll be more likely to be off than on. And lots of short negative elements will mean you'll have a default off state. They're more likely to be off than on. Right. Does it work? Ha! There are lots of outputs you can collect from this. So you can measure average, this is just an example run, measure average gene length, which turns out to be quite interesting, but I won't go into why. You can measure the number of expressed genes, number of producing transcripts, which turns out to be quite dull, and I won't say why. You start out with an entirely random genome, and it adapts fairly rapidly, and then sort of either plateaus or slowly increases adaptation. And you could look at the fitness, and that turns out to be really confusing, but I won't tell why. What I'm going to focus on is this thing here. This is, for each gene, pay attention now, this is complicated. For each gene, you look at the shortest positive and the shortest negative regulatory element. So that's the one most likely to be active. And then you say for all the genes in the genome, what is the average length of those short positive and short negative elements? If the short negative ones are shorter, on average, they're more likely to be on. That means the genes, on average, are more likely to be suppressed off. You've got a default negative, default off mode. If the positive ones tend to be shorter, they're more likely to be on. And so you have a default on mode. So, in this example, here's the average minimum regulatory element length, blah, blah, blah. Red is negative, blue is positive. The red is a little bit longer than the blue. There's a lot of noise on that because I had the mutation turned up too high. This is a great thing about doing life in computers. You can turn mutation up and down, things like that. So this suggests this is very weakly a prokaryotic default on mode, but it's pretty weak. It's pretty messy. I mean, you know, you wouldn't call that a good transit signal in an exoplanet lecture, would you? So here's a few more. Here's another one that's doing the same thing. I've just dialed down the mutation rate so it's less noisy, but it's weakly default on. I wouldn't say anything. This one can't decide what it's doing. It's just wobbling all over the place. But these two are more interesting. Here we've started to evolve a clear pattern, a signal of some sort. This one, the positive ones, these are the elements that turn things on, tend to be shorter. That suggests you've got a prokaryotic type default on genetics evolving. And this is the other way around. You've got the negative evolving. And you do this a lot for different combinations of parameters and different days of the week and things. And you try to correlate, are you getting default on or default off with all the different inputs? And this is what we've got so far. This is really preliminary. This is just straightforward correlation coefficient. Greater than, I think, 32 is significant at 95%, and those are colored in red here. And one really surprising thing that leapt out, I thought, it's going to be you put complicated environments in, and the genome's going to have to be more complicated to respond to it, and you'll have eukaryotic, and you'll have kangaroos evolving. It didn't care what the environment was. Wow, that really annoyed me. It was all about genome structure. The correlations that were more significant was about genome structure. And the thing that correlated with default off, evolving that sort of thing where you've got a nice curve separating like this, saying you've got a default off, was having a lot of genes. You could also alter the number of bases in your DNA in this too. You could say, let's have a 7-base genome. Let's see Steve Benner do that. Few bases, simpler genome, correlated with default off. Short genes. So this is simple genomes. It's the number of genes that correlated. Weird. So this isn't inconsistent with the original idea that having a lot of genes means it sort of favors evolution of this eukaryotic type default off genetics. It's not entirely supportive of it. It's about many regulatory elements, not their size or complexity. And this is why I mentioned the RNA world genome, because this sort of looks like that, doesn't it? Lots and lots and lots of RNAs, little short ones. And this hints that default off was the standard for first protolife. So, caveat, it's only a model, okay? I wish more astrobiology talks would say it's only a model, because quite often it is. It's only a model in Excel. I did this in Excel, okay? This is a bad idea. And I'm very fortunate that Enrico Borriello at Sarah Walker's group at ASCU is now recoding this into something that actually looks like code. So, conclusion. Genetic style tends to differ. We developed a model of that that's not completely abstract and yet not chemically specific. Initial results hint, and this is just a hint, that more genes mean a default off style. It's about regulatory group number, not about genome complexity. And that hints that this might be a primitive characteristic. In other words, you and I are more similar to Luca than E. coli. Your thoughts welcome, even yours. Because this is really preliminary and we want your thoughts and input before we go on to the next stage. Those are the papers, and one last call out, MDP as Life. I've published in Life several times, and so have one of my colleagues, and we published in another journal that you'll be familiar with that I won't name, that we publish in quite a bit in this context. And these guys are professional, faster, cheaper, and open access. So I'd have a look at MDPI's Life as a potential output for your next paper. I think you'll be pleasantly surprised by the level of professionalism and speed with which they can get your stuff out. Thank you very much.